There are three main stages to transcription: initiation, elongation, and termination respectively. During initiation, RNA polymerase binds to the promoter of the DNA, then the DNA strands unravel partially, allowing RNA synthesis to begin. Then during elongation, the RNA polymerase continues across the DNA in order to get an RNA copy. Nucleotides are added to the 3' end of the RNA molecule. Therefore, the RNA will be synthesized in the 5-3 direction. All 4 nucleoside triphosphates are required for this successful synthesis. Lastly, during termination, the RNA polymerase separates from the DNA, having synthesized a new RNA molecule. One strand of DNA is copied into a strand of RNA during transcription. Transfer RNA and ribosomal RNA is synthesized during transcription. Some genes are transcribed more frequently than others. Some genes are copied consistently, whilst others are only copied at a specific point in the life of the cell. Transcription factors are responsible for activating or inactivating genes. The transcription of DNA takes place in the cell nucleus for eukaryotes and in the cytoplasm for bacteria and archaea. Intermediate messenger RNA is used to transport genetic information from the nucleus to the cytoplasm.
Transcription in E. coli begins by firstly identifying the promoter sequences for genes from E. coli and also looking for consensus sequences because the holoenzyme needs to bind to a promoter. They are -35 hexamer, which consists of TTGACA, and pribnobox, which consists of TAT-AAT on the coding strand. These 6 base pair sequences are important for function of the promoter, therefore are highly conserved between species. They lie at positions -10 and -35 upstream of where transcription will occur. In E. coli, a single type of RNA polymerase makes mRNA, tRNA, and rRNA. It exists in 2 forms, the holoenzyme and the core enzyme. The holoenzyme is a pentamer that carries out initiation but not elongation. The core enzyme carries out elongation but not initiation. The holoenzyme binds to the promoter region about 40 to 60 base pairs in size, which contain the consensus sequences. The sigma subunit of RNA polymerase is needed for promoter recognition and therefore initiation of transcription. At this stage, the DNA is seen in the image as still double-stranded. With elongation here, the sigma subunit dissociates from holoenzyme to leave the core enzyme that continues RNA synthesis in a 5'3" direction using 4 ribonucleoside 5' triphosphates as precursors. Also, the DNA double helix is unwound and this forms a transcription bubble and is rewound after the complex has passed. Termination finally. A common termination signal is a hairpin structure that is formed by a palindromic GC-rich region, which is then followed by an AT-rich sequence. However, not all termination sites have this structure, therefore they need an additional protein called Rho to help recognize the termination site and stop transcription.
Introns begin and end with specific sequences in mRNA precursors. As introns are non-coding, they must be excised before transcription occurs. In eukaryotes, these sequences have a common motif, beginning with GU and ending with AG. At the 3' end of the intron, there is a stretch of 10 pyrimidimes termed the polypyrimidine tract. This tract is followed by a variant base and then C and the invariant AG. Another site within the intron is 20 to 50 nucleotides upstream, called the branch site, essential for determining where splicing takes place. Splicing consists of 2 sequential transesterification reactions. Several RNAs and proteins form a spliceosome and begin with cleavage of the phosphodiester bond between the upstream exon and the 3' prime of the intron. Attacking the group is the 2-hydroxyl group of an adenylate in the branch site. Between the 2 and 5 phosphodiester bond formed between A and the 5-terminal phosphate of the intron. It's important to note adenylate is also joined to 2 nucleotides by the 3,5 phosphodiester bonds. A branch is generated and a lariat intermediate is formed. 3-hydroxy terminus of exon 1 attacks the phosphodiester bond between intron and exon 2. Exons 1 and 2 join, and the intron released is in the lariat form. Again, this is transesterification. Splicing uses no ATP or GTP, as the number of phosphodiester bonds remains the same.
Initiation. The ribosome first dissociates into the 60S and 40S subunits. EIF2 prevents reassociation. Then EIF2 recognizes the first amino acids, always methionine in eukaryotes. The methionine and EIF2 binds to the 40S subunit. EIF4 recognizes the 5' cap, and the 40S subunit becomes activated. The messenger RNA bound subunit, 40S, will move along the messenger RNA until the AUG codon, which is methionine. This requires energy as well as other initiation factors. This process is called scanning. Once the start codon has been found, the 60S subunit binds to the 40S subunit. EIF5 is required for this and works by breaking down the bond between EIF2 and the 40S subunit. The methionine residue is now in the P site of the ribosome. Elongation is the process where amino acids are added to the chain. The order is specific to the sequence of codons in the messenger RNA. Amino acyl transfer RNAs are picked up by EF1. GTP is required for this step. This complex moves into the A site of the ribosome, where a methionine tRNA is present at the start of the protein. The anticodon of the tRNA is aligned with the codon on the messenger RNA. When the amino acyl tRNA moves to the A site, the rest of the peptide being produced is in the P site. A peptide bond is then formed by a condensation reaction. The newest amino acid then moves into the P site, which leaves the A site empty. The ribosome moves on to the next codon 3 bases along. As this happens, the amino acid in the P site moves to the E site. EF2 and GTP are required to move the amino acids from the P to the E site. These reactions will continue until the termination codon is reached. The process of elongation requires 2 GTP, EEF1, EEF2, as well as tRNAs and messenger RNA.
Once the stop codon is held in the ribosomal A site, the elongation process stops and the termination begins. A protein complex made of eukaryotic release factor 1 and eukaryotic release factor 3 recognizes and binds to the ribosome. This binding has the effect of cleaving the ester bond between the peptide and the tRNA, freeing the peptide for post-translational modification.
Proteins undergo many reversible changes, such as the covalent addition of a phosphate or an acetyl group. These post-translational modifications are used to regulate protein activity by inducing its conformation, its ability to bind to other proteins, and its location inside the cell. Most proteins are thought to be allosteric, meaning they can form 2 or more slightly different conformations. This is caused by the post-translational addition or loss of a ligand. In higher eukaryotes, the most common regulatory modification is the addition of a phosphate group. Protein phosphorylation is an enzyme-catalyzed transfer of the terminal phosphate group of an ATP molecule to the hydroxyl group of a serine, threonine, or tyrosine side chain. A protein kinase catalyzes this reaction, and a protein phosphatase catalyzes the reverse. A phosphorylation event can modify a protein in 3 key ways. Firstly, a phosphate group carries 2 negative charges. Therefore, its addition to a protein can cause significant conformational change, affecting the binding of ligands elsewhere on the protein. One way this occurs is by the phosphate's negative charge attracting positively charged amino acid side chains, affecting the protein's conformation and consequently its activity. However, this is a reversible change, therefore when the phosphate is removed, the protein's original conformation and function is restored. Secondly, an addition of a phosphate group can cause other proteins to bind to the phosphorylated protein, because the phosphate group can form part of a structure that other proteins recognize and bind to. One example of a domain that does this is the SH2 domain, which binds to phosphorylated tyrosine side chains. There are more than 10 common domains that bind phosphorylated amino acid side chains. Thirdly, the addition of a phosphate group can disrupt protein-protein interactions, as the phosphate can block binding sites between the two. As a result of these processes, phosphorylation and dephosphorylation often drive the assembly and disassembly of protein complexes. Eukaryotic cells also have another way to regulate proteins by the addition and removal of a phosphate, but in this case, the phosphate is not attached directly to the protein. It is instead attached to a guanine nucleotide, GTP. GTP binds tightly to the class of proteins called GTP binding proteins, which are also known as GTPases. Most commonly, GTPases are in their active conformation when GTP is bound. The protein is inactivated when GTP is hydrolyzed to GDP, causing the loss of the phosphate group. This reaction is actually catalyzed by the protein itself, as their name suggests. An example of a GTPase is the Ras protein. This protein has an important role in cell signaling. In its active GTP-bound form, the protein induces a cascade of protein phosphorylation within the cell. However, the majority of the time the protein is in its inactive GDP-bound form, only becoming activated when it exchanges GDP for a GTP molecule. This process is initiated in response to extracellular signaling, such as growth factors binding receptors in the plasma membrane, which causes a guanine nucleotide exchange factor to bind the Ras protein, causing it to release its GTP molecule, allowing GTP to take its place, as GTP is present in large excess over GDP. Ras is inactivated when it binds a GTPase activating protein, which induces the Ras to hydrolyze its bound GTP.
Acetyl groups can also be added or removed during post-translational modifications. Acetylation is the addition of an acetyl group at either the N-terminus of the protein or at lysine residues. This can occur with or without the assistance of enzymes. One example of this is acetyl transferase, which acts on the acetyl group of the acetyl coenzyme A and moves it to the side chain of lysine residues. The catalysts in this reaction are known as HATs, as they act on histones, but this may be changed to KATs due to lysine being termed K. The conversion of the positively charged lysine to acetyl lysine, like the addition of negatively charged phosphatases to neutral residues during phosphorylation. This causes different interactions with the proteins and can change the structure and function of the protein. Acetylation of histones promotes the arrival of effector proteins and relaxes chromatin conformation, leading to increased transcription. Like phosphorylation, acetylation can be reversed using histone deacetylases, known as HDAC, but may be termed KDAC.